So my goal today is to give an overview of multiplex imaging data and the different challenges that can be tackled by statisticians. So I'm not going to go deep into any kind of methodological area. I'm just going to give an overview of a bunch of different topics. For those of you who are at Emory, I'm going to be talking in the CBIS working group in a couple weeks, where I'll be giving a version of this talk, but I'll also be pitching a couple methods projects that I have available. So stay tuned for that if you're at Emory. Anyway, so here I go. And I'm going to talk about statistical analysis of multiplex single cell imaging data. Oh, and then the last thing to add is I really like to get questions as I give a talk. So if anything is not clear to you throughout, please feel free to jump in and ask questions. I'm happy to take them. Okay. First off, okay, what is single cell multiplex imaging data? I've got a cartoon of that here, but essentially it's high or medium dimensional analysis of tissue samples at the resolution of individual cells. So you take a slide, that's what this cartoon is showing here. You take a piece of tissue, you put it on the slide, and then you image pieces of that tissue. Usually circled, they're called regions of interest or ROIs. And then within that region of interest, you're able to see individual cells within the image that you get. So these little blue blobs here represent cells that are in the image. So single cell refers not to the fact that you're imaging one cell, you're actually imaging hundreds or thousands of cells in every image, but to the fact that you have individual cell levels.
I should be able to go now. Let me translate.
Okay. So then the multiplex part of single cell multiplex imaging comes in because multiplex refers to the fact that we're tagging multiple types of protein in the tissue for the image. So the way that it works essentially is you take these protein antibodies and you sort of attach them to the outside of this tissue. And the antibodies specifically attach to a certain type of protein. And each type of antibody has a different label on the outside that will then fluoresce in a different color when it's imaged. So red might be one type of protein, blue might be another type of protein. And multiplex refers to the fact that we have multiple types of proteins that are being attached to the outside of this tissue. So each protein label is then typically called a marker, and you have different types of markers. There are phenotypic markers, and these are used to define either tissue type, say tumor versus stroma tissue. This comes up a lot in cancer research, or cell type. So you have different types of cells in the image. You want to be able to learn what type of cells you have. And so the phenotypic markers will help you identify what cell type you have. But then there are also functional markers, and these are markers that inform what your cell is actually doing. And then these can be present across different types of cells. And then the imaging piece, which we know and love in this group, means that spatial relationships in the tissue are preserved. And the reason that this is interesting for this type of data is that precursor technologies didn't have any spatial information at all. Essentially, you took a tissue sample and then you grind up the tissue and put it into cell suspension. And then you figure out what cells are in that suspension, but you lose any of the spatial information about how cells are interacting with each other. So it's fairly new that we can get this level of information about the cells and also preserve their spatial information. So it's really cool that we're able to now do this. So putting this all together, you get these images, which are produced as multi-channel TIFF files, where each channel is a different protein marker. So if any of you have ever worked with RGB images, red, green, blue, those are TIFF files. It's very much the same thing, but an RGB image is a 3-channel TIFF file. With this type of imaging, you get an M-channel TIFF file, where M is the number of protein markers. So if you have 8 different protein markers, you're going to have an 8-dimensional TIFF file. And so each channel is a different marker, and each pixel contains a continuous intensity value for each marker in the image. So below is an example image from a non-small cell lung cancer data set that I've worked with. And this data set actually has 8 different marker channels, but I'm only showing 3 of them to the right here. So this image on the left is actually the composite image. This is what the image looks like when all 8 channels are stacked together. But each of these 3 are different channels. So this blue here is the nucleus channel. So this is called DAPI. It marks where nuclei are in the image. So each of these little dots is a cell nucleus. This is the same image, but now the red is highlighting beta-carotene, which is a marker for tumor cells. So the different red blobs here represent tumor areas of the tissue. And then yellow is CD8, which is a marker for a specific type of immune cell, which is something that we're really interested in studying with this type of data. So the yellow blobs, yeah, go ahead.
It just looks like Miranda Lynch has a question.
Yeah, I wasn't quite sure if we were supposed to interrupt with just kind of piping up, but when you're doing these sort of merges of the different modalities, do you have to do any re-registering of them, or are these essentially just, you know, snapshot after snapshot with no shifting of the spatial, you know, positioning of the things in the cell?
No, that's a great question. So we kind of think of them as being just one image combined. And there is a little bit of shifting that can occur, and there's spatial registration that occurs in the image collection step. But for the most part, it's relatively minor, and we don't have to deal with it as statisticians. Good question. So it's not so much the same as neuroimaging registration across modalities, where you have to do like sort of a huge warping of the brain in order to be able to align people.
May I ask a question as well, since you're on kind of this part of the talk? When you're actually putting these antibodies onto your tissue sample, is there any potential interaction, essentially, of the number of antibodies that you're putting onto the sample and what you're going to observe? Like, can you get some kind of saturation effect, even though they're targeting different proteins?
No, that's a really good question. So I would say there are 2 answers to that. There are a couple different ways that you can attach them. One is through fluorescence, and another is through, we're attach and then image them. For the fluorescence ones, if you have too many different type of proteins you're attaching at the same time, on the image, like the tissue end, they can attach pretty specifically to the protein. But on the actual visualization end, there can be overlap in like the fluorescence of the channels. And so it can be hard to deconvolute the signal and figure out which protein is attaching, like which protein you're actually imaging. So that limits the number of channels that you can have for one given, they call it staining, when you attach the proteins to the tissue. But one thing that people will do is they'll like stain the tissue with, say, 6 markers, and then they'll image it, and then they'll wash those off, and then they'll stain with like 6 different ones, and then they'll wash it off. And that can give you more information, but you can imagine with each round of like washing it off, some of the tissue degrades, so you get less and less good quality image. But there's,
Thank you.
but like that brings up a point that that's going to sort of come up throughout the talk, is like there is a ton of bias in this data, and a lot of pre-processing that I think that needs to be done and is underdeveloped in order to really trust the results you get at the final analysis step. But to your question, Miranda, yeah, please feel free to just like interject whenever. I love it. It makes the talk more fun for me. So I really appreciate the questions. So yeah, these are some great questions. Okay, so that's what your image actually looks like, something like this. Okay, so why are we collecting this data? So for the most part, at least for initial applications, people are collecting this type of data because they're trying to understand the relationship between immune cells in cancer better. So in cancer, this concept called the tumor microenvironment, which is the area within and surrounding a tumor, and it includes tumor cells, infiltrating immune cells of different types, blood vessels, and other tissue. And by better understanding the relationships between immune cells and the tumor, we can come up with better therapies that are personal and directed and sort of target specific interactions between those cells. And so one thing that there are a couple different questions that can be answered about the tumor microenvironment using this data. Some of the types of questions that I've seen consistently are what percentages of immune cells are present before and after chemotherapy in different types of tissue within the tumor, like tumor versus stroma areas. Or do patients with high spatial clustering of certain types of immune cells, which B cells and macrophages are 2 types of immune cells, do they survive longer? And that's a question that one of my students looked at a couple of years ago. And this is just a toy picture of the tumor microenvironment. So most of the questions that I've looked at are related to cancer survival and the tumor immune microenvironment, but you could use this for autoimmune diseases, or there are a lot of other potential applications as well. Really, if you have tissue, you can image it using multiplex imaging. So one thing I'm not going to delve into too much here, but is important to mention, is that there are several different technologies that are able to do this, and they've been quickly developing over the last couple of years. And they have different pros and cons. The different names of the platforms are here. I've worked mostly with Vectra Polaris data. So Vectra Polaris, I would say, is the most popular and widely used platform. And that's because it's been pretty well developed by industry and it's sort of the cheapest. And because of that, it's also been most published on up to this point in time. But it's also sort of the lowest multiplexicity. That's just the number of markers that you can get. You can only get about 8 markers with this instrument. There are other types of imaging, multiplex imaging modalities that can have as many as 40 or 50 markers, but the imaging processing pipelines for those are not as well developed. And I would say the data quality is often not as good. So there are trade-offs. And so a brief interlude, I want to give a comparison of spatial transcriptomics and single cell multiplex imaging, because these are areas that are similar in a lot of ways, but they're also still distinct. And I know somebody mentioned spatial transcriptomics, but I'm sure that others of you have probably heard of it because it's a pretty hot area right now. So both spatial transcriptomics and multiplex imaging are ways of getting spatial resolution information, a single cell resolution information about tissues samples, essentially. Spatial transcriptomics is a spatial analog of single cell RNA-seq, whereas multiplex imaging is a spatial analog of something called flow or mass cytometry. Multiplex imaging targets proteins, while spatial transcriptomics targets mRNA. Because transcriptomics is looking at genes, it can have hundreds or thousands of features per cell. So it's super high dimensional, whereas multiplex imaging is just going to have the number of protein markers that you can use, that you can image. So it's usually going to be, somewhere between 8 and 40 markers. So I would say it's much lower dimensional than spatial transcriptomics data. And for that reason, spatial transcriptomics is often focused on dimension reduction, but for multiplex imaging, that's not really a step that's required. However, multiplex imaging has some advantages that spatial transcriptomics doesn't. For example, it's typically much higher resolution. Like, it's truly single cell. You can see individual cells in the image. And spatial transcriptomics is some of the platforms are single cell. Some of them have roughly 10 cells per spot. And the data quality is typically much better for multiplex imaging. And multiplex imaging is cheaper, which means that you can image for tons more people than you can for spatial transcriptomics. So a typical spatial transcriptomic study will have maybe 5 subjects, whereas I have data sets of multiplex imaging with 400 subjects. And because of this, for multiplex imaging, you can really ask questions about patient-level outcomes and disease, and how the imaging features contribute to some patient-level outcome, whereas with spatial transcriptomics, we're not really there yet. Any questions about that? It's hard to say spatial transcriptomics too many times in like a 3-minute period. So I'm going to probably not say that again after this. Okay, so one thing about SCMI data that's super important to know is that there is a very complicated data and processing and analysis pipeline. So you're from imaging, you know that imaging data is complicated. That's true for neuroimaging data, and that's true for this type of data as well. But for this type of data, the pipeline is just not as well established yet because the technology is so much newer. So the first thing that happens is you acquire the image, then there's some image filtering and background correction. And then everything after that are certain areas where statisticians can have some impact. One I call statistical image processing, that includes image normalization, cell segmentation, and cell phenotyping. I'm going to get into what these mean a little bit more in the next couple of slides. But after filtering, you get your multi-channel TIFF file that I talked about. Then after normalization, segmentation, and cell phenotyping, then you end up with this nice table, basically a tabular data set like you could analyze easily in R, where each row is a cell, and you have features of the cell like marker intensity values for each of the marker, phenotype, which is the cell type label, what area of the tissue it's in, tumor or stroma area, and you also have XY coordinates, which is the spatial location of that particular cell. So once you have that tabular level data, then you can do your more typical analysis like compositional data analysis, which is what types of cells do you have, and spatial data analysis. And then finally, software development is really important in this area as well. Okay, so I'm going to talk a bit about statistical image processing. And Ruby, you will know about this very well. So Ruby, Simon's student, has been working in this area. And so those of you, and I know Andrew also works in this area for neuroimaging. So image processing and neuroimaging is sort of a hot area for statisticians. But it ends up being a little bit different in this area, though a lot of the concepts are the same. So with this type of data, you can work at the pixel level. So then you're working with the multi-channel TIFF files directly, and you're operating on pixel intensity values, or you can work at the cell level. So after cell segmentation, which I'll touch on what that means, then you have cells which are made up of pixels, and you can aggregate at the cell level, just take the mean intensity value across the pixels and operate with that cell level intensity value as well, which can be easier because you don't have as much data, your data isn't as large. So for cell segmentation, you have to operate at the pixel level, but for normalization or phenotyping, which I'm going to talk about after that, you can operate at the pixel or the cell level. I typically operate at the cell level, so that's what I'm going to talk about. But anything I talk about, you could think of applying at the pixel level as well. Okay, so let's briefly talk about cell segmentation first. So the point of cell segmentation is to identify cells and nuclei in the image. So the nucleus channel, you always have a nucleus marker present with this data. And that's used to identify where is the cell, which is important, where is the nucleus, which is important for segmentation. And then you typically have a marker of cell membrane or cytoplasm as well, and that is used to draw a boundary around the nucleus. So these little dotted red lines show the segmentations of the cells in this image. And then after that, you have sort of a cell object, and for each cell, it's made up of pixels, and each pixel has a marker intensity value for each of the markers in the data set. So there are a couple.
Jumping in with a question real quick.
Sure.
Are there separate markers for the nuclear membrane and the cell membrane?
So the nucleus itself, it's not the membrane actually. It's just the whole nucleus is usually DAPI is the marker used for that. But
Okay.
then, and then yeah, you have a separate marker for the cytoplasm.
Okay.
Or the
Thank
cell
you.
membrane. Yeah, good question. So there are 3 different buckets of approaches for doing cell segmentation. Some of them are sort of proprietary software that comes bundled with hardware that companies like Akoya, which have the most popular platform for this, give you. Usually they're GUI based and they're user friendly. They don't require training data, but I think their accuracy sometimes leaves something to be desired. Then there's also GUI-based open source software, which is also user-friendly and often doesn't require a gold standard. And I think that some of the groups I've worked with like to use these because they're a little bit more transparent in how they work. And then finally, unsurprisingly, the best methods for cell segmentation typically are based on deep learning. But they require lots of manually segmented training data, and they can be hard to adapt without computational expertise. And a lot of the people who are generating this data are biologists who don't necessarily have a whole team of like statisticians and computer scientists to help them clean their data. So they have to work with something like software, proprietary or the GUI-based software instead. So in this field, it's really like helpful to keep in mind the users of this data and making software tools that are super user friendly. But so what I would say, one of the state-of-the-art methods for segmentation is called Mesmer, and it comes out of a lab, I think at Stanford, and it's deep learning based. And they collected like just a whole bunch of labeled data from different multiplex imaging platforms and showed that their segmentation works well for a lot of different types of tissue and different types of diseases. So that's a good paper to check out if you're interested in implementing your own segmentation algorithm. Any questions about segmentation before I move on?
I just the manual work. Oh, I'm sorry. Yeah, TM.
Oh yeah, the manual.
That's very good.
Well, yeah, I'll ask my question, sorry. So that manual segmentation, especially for, you know, if you were trying to train up a deep learning algorithm, how long does that take to manually segment an image? How much manual labor is it?
Really, really painful. So actually
Okay.
the paper to go along with Mesmer talks about their manual segmentation approach too, where they actually like they built a GUI just for the manual segmentation, and then they paid a lot of people through like Amazon Mechanical Turks to do the segmentation, and they like trained them. So they have like a quality control check on the manual segmentation that was done by pathologists so that they know that they're training people properly to do like a cheaper version of the manual segmentation. So it's not easy. And it's because they have like a lot of resources that they were even able to like get a really large manually annotated data set.
Yeah,
So-
yeah, that's another thing I think needs to be emphasized is that you need a lot of manually segmented images, and you also have to worry about the rater agreement stuff for you know, and that's a whole separate analysis that needs to be incorporated. So I think there's a lot of issues with that, and I just wanted to kind of mention that.
Yeah, no, definitely. Cell segmentation is not an easy problem in this space, but it's also one that I think computer scientists have typically focused on a little bit more than statisticians. And I'll say here too that most of these images have fairly well-defined cell nuclei, and that's the nucleus is in purple. And a lot of the images I've seen are actually much messier. So the problem has been harder than they're showing here. So something to think about. So I'll move on to other aspects of statistical image processing.
Just 1 second. I'm sorry. I think Yao-Tian had a question,
I have
and I had
one
cut him
question.
off.
So what's the relative size about a cell and a pixel?
Related size of a T cell?
Yeah. No, I mean, what the size? Normally, one cell corresponds to how many pixels?
That's a really good question. I don't know off the top of my head. I think it's around like 5 to 10, but I think it depends on the resolution of your image and also what type of cells you're looking at, because some cells are a lot bigger than others.
Thank you. Then what about nuclei and the size of nuclei?
What about the nuclei? How big are they?
Nuclei.
Yeah,
Yeah.
that also depends on the cell. You have some cells that are relatively round, and you can just think about identifying the nucleus, which is easier than identifying the whole cell, and then just drawing the cell or like some radius beyond the nucleus. And those are easier problems, but there are also other types of cells, like neurons, for example, that don't have a round shape. So segmenting those is a lot harder because they have like little arms that go off into space, and it's hard to know where they start and end.
Thank you.
Yeah, no, that's a really good question.
One more comment, if I may. Dividing cells also have very atypical nuclei, and this is something that is in particular important if you deal with cancerous tissues. So that often requires taking sections from different directions in order to identify the frequency of dividing cells. So how do you deal with that?
No, that's a really good point. And I think that's something that isn't actually dealt with at all at this point in time. I think that people who are doing this analysis assume that there's a certain amount of error in the way that the cells are segmented, and hope that on average it sort of cancels out for the different types that they're looking at. But I think that being able to capture that would require a segmentation algorithm that is specifically trained for that purpose of identifying cells that are dividing. And big things like Mesmer haven't been trained to do that. So somebody would have to sort of train their own neural net to look specifically for those types of cells, which I think would be super worthwhile to do, but nobody's done that yet.
So 25 years ago, I already had a solution that the computer scientists are still working on.
What is it?
No, the sectioning in different directions in order to average out dividing cells.
Mhm. Nice. Okay, thanks a lot for your questions. I'm going to move on to the next section of image processing, which is intensity, normalization, batch correction, and harmonization. So hopefully this will speak to you, Andrew. But there's something with this data called the slide to slide problem. So tissue is placed on a slide and it is imaged. Each slide contains tens to hundreds of images, because each of these little boxes is actually an image. You're zooming in really close when you're looking at this data. But then you'll also image tens to hundreds of slides for each experiment. And you can imagine these slides aren't all necessarily imaged on the same day. They might be stained with antibodies at different times. There might be different tuning parameters for the machine that it's imaged on. And all of these introduce technical artifacts in the data that can mask biological effects, which we refer to these as slide effects or batch effects. Batch effects is common, in the larger genomic literature for describing non-technical or non-biological effects due to different types of experimental batches. So there are a couple different classes of techniques, transformation, normalization, and harmonization, meant to correct for batch effects in imaging data by adjusting intensities to remove those technical artifacts. So transformations are just things like log or square root transforms designed to make the intensity distributions look more normal, but they don't adjust for systematic effects. Normalization, I think of as adjusting the distribution of marker intensities within a slide to make distributions appear more similar across images. And batch correction or harmonization, I think of as explicitly removing bias across slides or across experiments. But all in all, they're all sort of meant to do similar things, which is remove batch effects. So I'm going to sort of refer to them interchangeably for this section. So, but these batch effects are a huge problem in multiplex imaging data, and one that's definitely not solved yet. The distributions of marker intensities can be very different across slides. And if you don't account for that bias, it can bias all the steps after that, including your cell phenotyping and your spatial analysis. And on top of that, they're actually usually not accounted for in analysis at all by most people who are analyzing this data. Unlike neuroimaging, where normalization and harmonization are well established, not necessarily which method to use, but at least it's well established that this is a problem that you should think about before analyzing your data. So this figure here I actually just made for a grant that's going in, and this shows some batch effects in 3 data sets that I have of multiplex imaging data. And it's just for one particular marker of a certain type of immune cell. The different colors represent different sites, and each little box represents a slide. And so the mean values are very different across slides. And the variance is different across slides. And this is also different across different percentiles of the intensity distribution. So we are seeing huge batch effects here, and these need to be accounted for. There are some reasons that you can't just take methods from neuroimaging and just apply them directly to multiplex imaging data. And that's because the distributions of the marker intensities look very different. They're highly right skewed, They're also typically zero inflated, and they have overlapping intensity distributions for what is considered positive or negative for a marker. And also slides differ in composition from one to the next. So you can't like spatially register results from one slide to the next, in the sense that you don't have like this part of a brain can be registered across slides. That's just, there's no correspondence in that way. And on top of that, imperfect cell segmentation, which I just mentioned in relation to your questions, causes some uncertainty in the marker intensities. And there's also a lack of ground truth to evaluate whether your normalization is even good. So there are a lot of compounding problems here that make it difficult to do batch effect correction in this data, but it's still super important. So Ruby mentioned Simon Vandekar, who's a collaborator of mine and her advisor. And so one of his students who graduated a couple of years ago came up with a batch correction evaluation framework for this data. And he essentially, oh, Simon's here. Hi, Simon. Hi, Simon. And he came up with a couple different metrics for evaluating whether or not your batch correction is method is good in the absence of a gold standard. One of them is a metric that measures alignment of marker densities. Another is cell phenotyping discordance, which in the interest of time, I'm going to skip explaining what that means, but it's a nice metric and I like it a lot. And I refer you to this paper if you're interested in learning more. And another is proportion of variance due to slide. And he also evaluated 9 combinations of transformation and normalization methods to figure out which one worked best for the multiplex imaging data he was working with based on his metrics. And he found, he actually tried combat as one of the methods, and he found that mean division typically worked best, and it's pretty simple. And on top of that, Cole came up with an R package that you can use to implement the metrics that are in his paper. And what's really nice about his R package is it allows you to use all of the default normalization options from his paper, but you can also specify your own normalization options. And you can specify them, and then it will calculate the metrics for evaluation so that you can determine whether or not your new method is better than the previous approaches that he tried. And I would refer you to that package to check it out. It's really cool. Any questions so far? Okay, so that's it for batch correction.
Sorry for interrupting. I have a question about normalization. So you mentioned that it is helpful to normalize within an image, but what if we have different slides and we try to normalize one channel for different slides, not within an image? Would that be helpful to get rid of the artifact effects, or use one threshold for the whole slides that we have, for example?
Yeah, so I think maybe that got a little washed out in the way that actually that's exactly what this metric is meant to capture. So we are trying to normalize the channel across the slides, and the unnormalized data typically has very different distributions of the marker intensities across slides. And sort of our assumption, Simon and Cole's assumption in developing this metric, is that if you have good normalization, then your marker intensity distributions within the same slide or across slides should look relatively similar. And that's what this metric is just attempting to capture.
Okay, thank you.
Julia.
I had a question on this too. Oh, go ahead first.
No, okay. Since you're actually breaking a tissue sample up into many small individual images, I would imagine that certain images have more or fewer of certain types of cells, right? With my biological understanding here is not great, but different types of proteins would be more prevalent in certain images. Would that affect the normalization? Because you might expect higher intensities in certain images than in others, depending on the composition of cells.
No, that's a great point. And I wish I had a histogram of the intensities. Okay, so the cell phenotyping slide, which I'll get to in a couple slides, will sort of help answer this. This shows distributions of the same marker for across different images. And what you typically do with these marker intensities is you draw a line that says, okay, to the right of this line, you're positive. Like all of the cells to the right of this line have this with this value are positive for that marker, and all to the left are negative. So you might have more cells that are positive, that have higher values in an image that has more of the cell type, but you would still expect or hope to see that the distributions themselves were fairly similar. Because if you imagine, say, this blue line was shifted to the right like 3 units, then it would be hard to tell from the blue line versus the red line where positive is, if you wanted to make a designation of what positive should be across the different cells. I guess another way of saying that is the intensities don't have any kind of inherent unit. So by doing normalization, you're helping to make them more comparable across images and figuring out like what counts as marker positive. Does that make sense?
Sorry to interrupt also, but just to clarify, so you basically have features measured on a cell level, right? So it's like after segmentation, each cell has a certain normal. Okay, got you. So I guess I was wondering too, so like, I guess the slide has background where there wouldn't be expected to be any cells. Are those different between slides as well?
The background, so typically these slides are background corrected, which means that the background is subtracted, like areas where you don't think that you should have intensity is subtracted from the whole image.
Oh, I see. So the background's already being used kind of in a harmonization-like approach.
Yeah, that's typically like sort of a pre-processing step, but as a sort of Simon and Ruby have found, that creates like an artifact of a spike in zeros due to the background correction in some of these images, which has to be then dealt with when you're doing the cell phenotyping.
Got you, got you. Okay, that's good to know. And sorry, just talk about even farther. I'm a little curious. Wait, so are these images acquired at like a single depth, or are they kind of multiple images across depths that are aggregated together to be one 2D image?
Different depths, you mean like, so they're not 3D if that's what you're asking. Like you'll just like one tissue slice and treat it as 2D. Is that what you mean?
Oh yeah, I was just curious because like a lot of microscopy like will involve like different depths of imaging, and then that they like kind of collapse it together. So this just acquires at one depth, just a single slice.
Just like C slices?
I'm not sure actually. So it's like, it's just one tissue sample. So you don't typically do like different layers of tissue and get the full like 3D picture of the tumor. But in terms of the actual imaging, how it's collected, I'm not exactly sure.
Okay, got it. Okay, well, thank you so much.
Yeah. Good question. Okay. These are great questions and I'm having a lot of fun and I'm probably not going to make it through all my slides, but that's okay. I'm happy to share them if people are interested in reading the ones that I don't get through for after this. Okay, so the last step in image processing is the cell phenotyping step. So that's your cell type labeling. And this is the process of identifying cell types from marker expression values. So essentially you have marker intensities, say each of these represents the distribution of marker intensities for cells in a given image. And conceptually, the goal is to create a cut point in that marker intensity density where cells are either positive or negative for a marker. And you can imagine this is relatively easy to do if you have a nice bimodal density like this blue one here. You can just draw a line somewhere in that trough of the density, and then all cells that are have values to the right are positive, and all cells that are to the left are negative for that particular marker. And so the way that we actually do phenotyping from a biological perspective is you'll have different markers. And then if markers are positive or negative in certain combinations, that will tell you what type of cell you have. So for example, a T lymphocyte, which is an important type of immune cell, will be positive for CD45 and positive for CD3, which are two different types of protein markers. However, with this type of data, you often don't get clear bimodality like this blue line here. What's more common is you might have a combination of lines like the ones here. This is real data that I'm showing you here, where you have bimodality for some of the different images, like this blue one, this purple one looks generally okay, but then you'll have a lot of images like this green and this red one, where it's really unclear where the values should be considered positive because you don't have bimodality. Like this red one, for example. And there are a couple different approaches to doing phenotyping. One is marker gating. And this is, I think, the approach typically taken by biologists in the past, where you just look at every single image or a collection of a couple different slides, and you just look at the marker densities and you just say, okay, I think that marker positivity starts at 1.5, and you just draw a line there. So that can be very manually intensive, and also not necessarily, it can be a little bit arbitrary. So as statisticians, we try to develop more automated approaches. Unsupervised clustering methods have been popular. Seurat and Phenograph are some that have built-in software. And most of these have been developed for other types of single cell analysis, not necessarily single cell imaging. And in those types of data modalities, the distributions tend to be more bimodal and less noisy. So those methods don't often work well for this data. And then there are also semi-supervised approaches. And there are a couple listed here. There's one developed by some colleagues of mine or former colleagues at Colorado. There's a steer, and then there's Gamma Gator, which is a brand new approach that was developed by Ruby and Simon just recently. And I think that their approach is really cool. So you should check it out. And they have a brand new sticker, which is awesome. Okay, so I'll get through as much of the, actually, do I have any questions about this phenotyping or batch correction before I move on? Okay. So finally, we get into statistical analysis. And so at a high level, we want to know how these multiplex images relate to patient level outcomes. So the first step of that is to quantify characteristics or features of the image. Features can be like cell type proportions or degree of cell clustering in the image. You extract these features and then you relate them to your patient level outcome. Those features become model covariates, and outcomes are typically things like survival or tumor subtype or disease progression. And a former student of mine, Ben Steinhardt, developed like an interaction metric for this paper that we published a couple of years ago. So why can't we compare these images directly like you can in neuroimaging? Well, the images are highly heterogeneous. There's not really correspondence across images. You don't have like a brain, for example, in each image. So you can't do spatial registration. The only real way to compare across images is to extract features from them. And so cell type proportions are usually something that the first line of attack people want to look at, but they don't incorporate any spatial information typically. So I think it's a little bit more interesting to extract spatial features and then use those in a model. So I'm going to dedicate the rest of the time to spatial analysis. So features that you can extract from these images are called spatial summary statistics. And they're used to describe the spatial relationship amongst different types of cells in an image. And often we separate by tumor and stroma. You want to look at clustering of, say, immune cells in the tumor regions and in the non-tumor regions. There are univariate summary statistics, those that look at clustering or dispersion of cells of one type. And there are also bivariate statistics, which look at co-expression or co-clustering of two types of T cells. And a lot of methods, a lot of popular methods are based on spatial point processes. They can analyze, say, the number of neighbors. These are the Ripley's K function and L function are two examples of that. Or you can analyze the distance to the nearest neighbor. And G function is an example of that I like. So if you're not familiar with spatial point processes or spatial point patterns, Point pattern is just a distribution of points in space. And in the context of this data, a spatial point pattern would be the distribution of cells in an image. And the locations of those cells are considered random and to follow a point process. Marked point patterns have covariates attached to each cell. For example, that cell shape or area or expression of a certain marker. And multi-type point patterns have multiple types of points. So typically we're working with multi-type or marked point patterns. Multi-type point pattern would be a point pattern where you have multiple types of cells. So the multi-type would be different types of cells. What I have here is an image where the red cells are immune cells and the gray cells are tumor cells. And we might be interested in the spatial arrangement of immune cells in the tumor. Here's just another example of a spatial point pattern of multiplex imaging data. In this example, the green cells are in tumor tissue area, the pink cells are in non-tumor tissue area, and then the black cells are macrophages, which is a type of immune cell. And oh good, Ruby is sharing the sticker, you should definitely check it out. And you might be interested in the clustering of macrophages, both in the tumor areas and the non-tumor areas. So typically what you are interested in is detecting whether or not you have more clustering of cells than you would expect if your cells were randomly distributed in space. So I'm going to skip through a couple of the lines of the theory about that and just show you a picture. Here we have, say we're interested in the red points, they represent immune cells. These are randomly distributed immune cells in this image here. And then the one totally to the right, we have clustered immune cells. And if we look at Ripley's K, that's a spatial summary function of those cells. The blue line represents random distribution and the black line represents the empirical clustering metric for those cells. And if the black line is above the blue line, that means that you have clustering. So in this image, to the left, there's no clustering, it's right around the blue line. And then in the image to the right, we have clustering, and you can see the black line is well above the blue line, which indicates that we have clustering. So what you can then do once you have these spatial summary metrics extracted is you can use them as covariates in the model. And I'm going to skip most of the rest of this now, just out in the interest of time. But there's some cool stuff you can do with these spatial summary functions using functional data analysis, which is one of my areas of expertise that we're going to put out some more papers on in the near future. And I'm happy to share these slides. But what I really wanted to get to here are some resources that I and other people have put together about multiplex imaging data. One is the Vectra Polaris data package, and this is a publicly available data set with 2 large experiments that I got from collaborators when I was at Colorado. And you can just download this directly off of Bioconductor and play with it yourself. Yeah, there are two full data sets with over 100 people in each data set with like patient level outcomes like survival, and also over 1 million cells in each data set. So those are a nice resource if you're trying to get sort of understand and get used to this data. There's also a short course on multiplex imaging that I gave at Michigan back in April. And there are slides and code that it's like fully reproducible up there if you want to do a little bit more with learning this in R. Simon and I also wrote a textbook chapter on this, which is on my website. And also Simon runs a national working group for this. So feel free to email him or me if you're interested in being a part of that. Cool, that's it. I have a lot of collaborators and they're great.